rotations. MALT stands for mucosa-associated lymphoid tissue, MALT. And the mucosa, of course, refers to the tunica mucosa. So instead of saying tunica mucosa, we'll just call it the mucosa. Or instead of tunica submucosa, we'll just call it the submucosa. So it's just a shortened reference to the tunica mucosa. The tunica mucosa actually starts in the oral cavity. The tunics are, not uniform in the oral cavity, but it is basically a tubular entrance into the digestive and respiratory system. So it does have tunic terminology. That's where you find the tonsils. They're located in the pharynx. Some of them are in the oropharynx and some of them are in the nasopharynx. You will learn about that in gross anatomy. In histology, what do we want you to do with these tonsils? We want you to recognize them when you look at them, and we're going to give you some clues on how to recognize them. And we want you to understand what their function is, their basic function is, and you'll learn more about them if they want you to know more about them in your immunology course. Okay, so let me move the notes out of the way. There are 3 examples of MALT that we want you to look at today. 2 of them are tonsils and 1 of them is gut-associated lymphoid tissue. So MALT has 2 subcategories. It has GALT, which is gut-associated lymphoid tissue, and then it has BALT, which is either bronchial or bronchiolar associated lymphoid tissue. You saw that with the bovine lung section. That lung section was a beautiful example of BALT, bronchial and bronchiolar associated lymphoid tissue. Here in the gut, one of the subcategories of MALT, we have tonsils, and then we have what are called Peyer's patches in the small intestine. So we're going to look at tonsils. There are 2 different kinds of tonsils that we will look at. This is a palatine tonsil of a carnivore, and the other example is a tonsil from a horse. It's called a cryptic type tonsil. I'm trying to think, it could have been lingual, it could have been palatine, but probably was palatine. And it is, again, an example of a cryptic tonsil. So what do we mean by crypts? Crypts are invaginations from the surface that increase the surface area. So this is our surface epithelium. So we're talking about oral cavity. So what type of epithelium lines our oral cavity? Okay, so what does it look like here? Well, this is stratified squamous epithelium, and then you would need to tell me whether it was cornified or non-cornified. Okay, so this is non-cornified, stratified squamous epithelium. It's got a really dense underlying lamina propria here. And then deep to the lamina propria is where we find our immune tissue. Now, invaginations from that surface epithelium are like if this was some yeast bread dough in a bowl and you poked your finger down into it, and when you pulled your finger back out, that indentation where you poked your finger stayed, okay, like a little tunnel that communicated with the surface of the dough. That would be an invagination from the surface, and you carried that surface of the dough in to that invagination when you poked your finger down in it. So that surface epithelium extends down into these invaginations, into these crypts. So these crypts are lined with stratified squamous epithelium. Now you might be saying to yourself, that is some of the oddest stratified squamous epithelium I have ever seen. And I would have to agree with you on that. Why is it so odd? Because we've got lots and lots of immune cells trafficking through or crawling through or passing through the intercellular spaces and even passing on into the lumen, okay? So that's why it looks odd, because you've got a lot of lymphocytes crawling around in that epithelium. Now, what did we learn about the organization of the lymphocytes in secondary lymphoid organs? This is definitely a secondary lymphoid organ. We learned that the organization of the lymphocytes was nodular and diffuse. And the nodules were what lymphocytes? They were B cells. So here's a B cell nodule, here's a nodule, there's a nodule, everywhere's a nodule, okay? Now, where is the diffuse? Well, just like the paracortex in the lymph node, the diffuse is everything that fills in around the nodules, and that would be your T lymphocytes, okay? So here's the diffusely structured lymphoid tissue, and that again is heavily populated with T lymphocytes. The nodules, the more organized lymphoid tissue, that's the B cells. Now, the diffuse lymphoid tissue often extends up into the stratified squamous epithelium. It's not doing it in this cryptic tonsil. Interestingly enough, I don't know if you've ever heard of tonsil stones. You've probably had them yourself. You have cryptic tonsils. Your palatine tonsils are cryptic, and meaning they have these invaginations, these mucosal invaginations. And you can see right here that the stratified squamous epithelium is doing what that epithelium does. It sloughs the squames, the surface cells that have died, and they are accumulating in here. Plus, we've got a lot of dead lymphocytes and even granulocytes, neutrophils in here. And it's forming a concretion, and this concretion can actually become mineralized. And ever so often they'll dislodge themselves or work their way out, and they're called tonsillar stones in people. I don't think we call it that in animals, but I just thought, you know, you might find that interesting. Okay, so that's the equine palatine tonsil. You don't have to tell us that it's a palatine tonsil. You just need to recognize it as a tonsil, know that it's a cryptic type of tonsil, and tell us that it's got diffuse and nodular lymphoid tissue. The carnivore tonsil has got a little bit more additional information to help you know it's a tonsil. The key in recognizing a tonsil, the key is always the stratified squamous epithelium. And don't think, well, that's a weird and unusual stratified squamous epithelium, I'll never recognize it. We'll just drive around. Here's fairly normal looking stratified squamous epithelium right here. Again, it's non-cornified. And then if you drive around, oh, wow, look here. That's where the diffuse lymphoid tissue, looks like a lot of T lymphocytes, are migrating up into that stratified squamous epithelium. You can even see some dead granulocytes here in those intercellular spaces. Those will be sloughed as these squames are sloughing. Well, here you can see some squames sloughing right here, and you can see these dead granulocytes and lymphocytes sloughing, mostly granulocytes that die and slough. Lymphocytes are more long-lived. Okay, so stratified squamous epithelium, that's key in IDing tonsillar tissue. The second thing that's key is recognizing that you've got both nodular and diffuse lymphoid tissue. So if you see nodular and diffuse lymphoid tissue capped with stratified squamous epithelium, you're done. You know that it's a tonsil. You might also additionally see some salivary glands. Okay, you might see those depending on how deep the section, how much of the surrounding tissue was made, you know, how deep the cut was made when we removed that tonsil from the cadaver. Also, you might see some skeletal muscle, and these would be pharyngeal swallowing muscles here, which we'll learn about those in gross anatomy. And this is skeletal muscle. You can see those peripherally located nuclei. You can see the little triangular shaped myofibers and the stippling of the myofibrils inside the cell. Okay, so that's accessory extra information to help you ID the tonsil, but really all you need is diffuse and nodular lymphoid tissue capped with stratified squamous epithelium. Are these primary or secondary lymphoid nodules? Well, I'm glad you asked. So these are secondary. You see the mantle here, the darker staining mantle and the lighter staining germinal centers. So these are secondary nodule. Just FYI over here, this is way cool. This is a dog. So, you know, they're always chewing on sticks and things they shouldn't be chewing on. So he probably stabbed his tonsil with a piece of stick or something. We've got a little granuloma right here, a little bit of pathology going on over on the periphery of that tonsil. Kind of cool, huh? You'll learn about stuff like that in pathology. Okay, down in the small intestine, we have gut-associated lymphoid tissue down there too. Now, in neonates of some species, ruminants, carnivores, and pigs, these pyre patches, true pyre patches, are form in the neonate, and that is where B cells differentiate and mature. So that would be a primary lymphoid organ in the neonate, along with the thymus. Now, after the neonate, after the fetus is born and is no longer a neonate, it grows up and becomes an adult, then that Peyer's patch actually goes away. And for future B cell formation, that would come from the bone marrow in those animals. But we do get aggregates of diffuse and nodular lymphoid tissue in adults, all adults, even humans, even these animal species that had true Peyer's patches when they are in the neonatal stages. But as adults, these Peyer patches can form anywhere. It tends to be the small intestine. And we still call them Peyer's patches, even though they're not true Peyer's patches. We're all just lazy and we just call them Peyer's patches, but they're actually secondary lymphoid organs at this point. And so what does that mean? Well, they're not just nodular as they would be with a true Peyer's patch. They now also have diffuse lymphoid tissue. And that's what we see here. And we'll learn more about this tissue. We'll revisit it again a bit when we're in the digestive system. So these are villi up here. This is part of the mucosa. This is our muscularis mucosa. So that lets me know I'm down here in the submucosal region. So that's where the bulk of these nodules are located. These are, let's see, are they primary or secondary? It's kind of hard to tell. Most of them are fairly diffusely, densely stained. So that would be a tough one. I probably wouldn't ask you on an exam whether these are primary or secondary because it's kind of tough to tell. There does seem to be a lot of macrophage activity. We have all these little punched out spaces here. So we know we've got a lot of active macrophages, but I don't really see a well-defined germinal center. So we won't worry about whether they're primary or secondary. Do know that they're nodular, and so therefore that's where B cells are located. Do recognize that the diffuse portion is the T cells. They tend to be just under the lamina muscularis here. They'll also kind of be in between the nodules, but mostly they'll be in this area between the nodule and up to the muscularis mucosa. In some areas, you'll see they break through the muscularis mucosa and obliterate it. We can't even see the muscularis mucosa right here. They've just spread those muscle fibers apart as these lymphocytes are passing through up into the lamina propria of these villi. These are mostly T lymphocytes, and they will also pass into the intercellular spaces of that epithelium. There's something called a microfold cell. We won't worry about it. We won't try to find it. The microfold cell is just a particular antigen presenting cell that is monitoring the luminal contents for foreign antigens and presenting those antigens to the underlying T cells and macrophages and dendritic cells, the cells that will have antigen presenting, further antigen presenting function, cells that might take that antigen down to the nodules to present to those B cells. So the microfold cells, you would need an electron microscope to identify or a special stain. We can't see them. But look how right here in this simple columnar epithelium, we don't have goblet cells right here. That lets me know that we've got microfold cells in this area, and that's where they're monitoring the luminal contents of the small intestine for antigens that they can process and present to these underlying lymphoid cells. All right, so that's really all you need to know for wrapping up the immune system. Now we're going to start the nervous system. And I know that tends to make people nervous, no pun intended. It's just a brief introduction today, and I think it's a good way to ease into the nervous system. We'll have central nervous system tissues, we have peripheral nervous system tissues, and we have autonomic nervous system tissues. Today, we're just going to focus on the central nervous system, which would include the brain and spinal cord. And really today, we're only going to focus on the brain, specifically the cerebral cortex and the cerebellar cortex, with a little bit of the underlying white matter. There's a whole lot of other information that we'll learn when we continue the nervous system in our next lab. We'll have 2 hours of nervous tissue at that point. Right now, we want you to think about white matter and gray matter. We want you to know that in the central nervous system, the white matter is deep and the gray matter is superficial. And the gray matter is what we call the cortex. So if we're looking at the gray matter of the cerebrum, it's the cerebral cortex. If we're looking at the gray matter of the cerebellum, it's the cerebellar cortex. We want you to recognize that gray matter, recognize the white matter, tell us a few things about both the gray and white matter, particularly in the cerebellum, where it's got 3 really easily distinguishable layers to that gray matter. And we'll look at some of the meninges. There's going to be a little bit of the meninges present, not all of the meninges. We'll meet the rest of the meninges when we look at the spinal cord. Let's see, going through the notes here, what else do we need to know? Oh, there will be glial cells. There are peripheral glial cells and there are central glial cells. What the heck are glial cells? Glial cells are cells that support the nervous tissue, so they support the neurons. In the central nervous system, there are 4 different types of glial cells. We'll meet most of these in the spinal cord. Today, we will look at some oligodendrocytes. Now, these glial cells, and we'll look at ependymal cells. These glial cells tend to require, at least the microglia, oligodendrocytes, and astrocytes, they really kind of require a special stain to see the entire cell. And that's true, really, in the nervous system. Why is that? Because in the nervous system, particularly the gray matter, we don't have a lot of, we don't have any myelin in the gray matter, and we don't have a lot of connective tissue. And all of the cytoplasmic processes, and I mean there are a lot of cytoplasmic processes in that gray matter. There are dendrites, there are axons, and there are lots and lots of nerve cell bodies and the cell bodies and cytoplasmic processes of all these glial cells. And they all just kind of mesh together. And the resolving power of the light microscope is not high enough to visually distinguish the boundaries between all these different cytoplasmic processes and cell bodies. So we have a term for that. We call that network, that mat, like a felt mat, we call that neural pill or neural pile. It's actually okay to pronounce that either way, neural pill or neural pile. And that's just that matted network of all these cytoplasmic processes and cell bodies that we can't see the spaces between. And so it all looks fairly uniform to us. So we just reference it as neural pile or neural pill. So we won't see the cell bodies of most of the support cells. We will see the cell bodies of some of the neurons because they're so much larger. The support cells, therefore, we tend to identify just by their nuclear characteristics, the shape and size, and the chromatic characteristics they have, and we just make educated guesses as to whether we're looking at an oligodendrocyte or an astrocyte or a microglial cell or an ependymal cell. Some of those educated guesses are pretty educated because we know where these cells hang out, where they live, and what they do for a job. The meninges, you'll meet all the layers on the spinal cord section. Today, you'll only be meeting the pia mater. And here's what you need to know about it. It is tightly adherent to the surface of the brain and the spinal cord. It's a very vascular layer, so expect a lot of blood vessels. And it's got just a little bit of connective tissue. So with a trichrome stain, you will see that blue stain of the connective tissue. So let's take a look. This is a section of a rat cerebrum. Now, the rat is not like the domestic species. It's not like humans. It doesn't have a lot of folds on the surface of the cerebral cortex. So this is the cerebral cortex from here to here on the rat, okay? And it doesn't have the outward projecting folds, which are called gyri, and the inward projecting folds, which would be sulci. It's just a smooth cerebral cortex, and there's a name for that. They're called lissencephalic. You don't really need to know lissencephalic. That's just a really cool word that lets you know they have a smooth cerebral cortex instead of one that has gyri and sulci. we've got underlying thalamus and hypothalamus and brainstem, and you don't need to worry about all of this in this region. We're just interested in that cerebral cortex. So let's zoom in a little bit more from here to here. Here's the underlying white matter. Here's axons. So cortex refers to gray matter, okay? So the gray matter is from here on this at the surface down to where you start seeing the white matter. So if I drew a line here and then I drew a line here, and that bracketed area right there would be the cerebral cortex. Now you can see the pia mater is peeling off right here. It's a very tender membrane, very fine. In fact, it's called the tender mother, the pia mater, pia tender and mater mother. And therefore it peels rather easily. It's a sectioning artifact, and you can see that it passes down into this this little sulcus right here, this groove in between the two cerebral hemispheres. And if I zoom in, you can see that it is indeed highly vascular, this pia mater. Okay, with just a little bit of connective tissue, the tender mother. Now, if we zoom out, you can see that the cerebral cortex has an organization of neurons. And neurons can be many different shapes. They can be pyramid shaped. Those are called pyramidal neurons. So this pyramid, these are pyramidal neurons. They can be round. These are granular neurons. And you'll see that I'm really just referencing the nuclei on these neurons, a little bit of the cytoplasm here on this pyramidal neuron, but here on this round neuron, that's just its nucleus. That's its nucleolus. I can't see much of its cytoplasm. It's just disappearing in this background neuropile that we've got right here. And that's not uncommon in the central nervous system. Sometimes you'll see some of the cytoplasm, like right here with this round neuron, and sometimes you won't, like here, we just see the nucleus, okay? Now we aren't going to learn all of the layers, there are different names for all these different layer organizations of neurons. There can be multiple layers, 6, 9 different layers, depending on the species and the region of the cerebral cortex. So we want you to just know that this is all gray matter. And look how right here, this outer inch or two, we don't really have neurons here. We just have the cytoplasmic processes, and some support cells. So we're going to call this the molecular layer. We do want you to know that. That's that name of that outer layer that doesn't really have many neurons at all. It's got some blood vessels, tiny little blood vessels. Here's a vessel, here's a vessel. And it's really just got nuclei of support cells, those glial cells, okay? So that's the molecular layer. And the rest are all these different neuronal layers that we're not going to distinguish and name. Now, one of the glial cells, the support cells that you'll see in the gray matter is the oligodendrocyte. The oligodendrocyte is interesting because in the white matter, it performs a myelination function around the axons, but in the gray matter, it forms a support function to the neurons. And here's one right here, this oligodendrocyte, this glial cell, and it's its nucleus. We don't see its cytoplasm. Here's a round neuron, and see it's right here where we call these perineuronal satellite cells. You know, they just live right within orbit, if you will, like a satellite of these neurons, and they provide a support function to these neurons. So here's an oligodendrocyte nucleus. Here's one, here's one, here's one. How do I know that? Well, it's one of the easiest glial cell nuclei to recognize. I call this thick nuclear membrane its eyeliner. So it's got this very characteristic thick nuclear membrane with a characteristic euchromatic interior to that nucleus. So it's a very easy cell to identify, the oligodendrocyte cell nucleus. Now, if you come down here in the white matter where these oligos are performing a myelination function, you'll see a lot of oligos down here. Here's a whole cluster of oligodendrocyte nuclei. Sometimes I call it blowouts. They kind of have blowouts when we process them, and that's just, there's not a lot of nuclear detail there. I just call that a blown out oligos right here. So don't pay any attention to that. That's artifact. Something went wrong with the preservation of that nucleus. But here you can see some well-preserved oligodendrocyte nuclei. And this is white matter. These are axons running left to right, so we see them in longitudinal profile, and then these are axons coming out of the plane of section towards us or away from us. And so they are in transverse profile. Here's a beautiful blood vessel with gorgeous little red blood cells in it that look like little pieces of a pomegranate. Okay, one of my personal favorites is the cerebellum. Why is it my personal favorite? I love tissues that you can ID from across a room. If I saw this standing as far back as my aging eyes can tolerate, then I would still know it's the cerebellum because it's beautiful and nothing else looks like the cerebellum. And it's my friend because nothing else looks like it. And therefore, I know it when I see it, like a good friend. The cerebellum has folds and sulci. Its folds are called folia. I don't know if you've ever heard the cerebellum referenced as the arbor vitae or the tree of life. So the tree would have foliage. So folium. This is one folium, plural would be folia. So we go up and down into a sulcus and then come back up and down into a sulcus. So from this sulcus to this sulcus right here, there's not a lot of space in that sulcus. Then this width right here would be one folium. So this is a folium right here. Now these folia give the cerebellum a lot of increased surface area. So you see our molecular layer out here. Yes, the cerebellum has a molecular layer. That's that region from under the pia mater, here's the pia mater, down to where you first start seeing neurons. Okay, so this is that molecular layer that's just filled with neuropile and glial cell neuronal nuclei, glial cell nuclei, and a few neuronal nuclei here in the cerebellum. But you'd have to do a special stain to really appreciate these neurons. Here's some neuronal nuclei here, probably basket cell neurons, but you'll learn about those in neuroanatomy. Okay, so what we want you to recognize is this pia mater here. I want you to know that we're looking at a folium. We're looking at the molecular layer here. These nuclei are glial cell nuclei. We've got a lot of blood vessels in here. What else do we have? Well, under that folium, and see, because under that molecular layer, and because we have folds or folia, here's that molecular outward projecting folium and then coming back down, lining that sulcus of that first folium. And then we're going to take a hairpin turn here. And ooh, it says my internet connection is unstable. That's not cool. And then that molecular layer is coming back up, bending around and coming back down again. So that's another folium. Now we have, just like the molecular layer makes a bend with that folium, so do these other layers. Next in line, we have the piriform cell layer. Piriform cell means pear-shaped. So these are pear-shaped neurons, very large. You probably have heard them called Purkinje cells. That's okay, but Dr. Purkinje named a lot of things after himself. So this isn't the only thing he named after himself, but this is one of them. You can call them Purkinje cells. We like piriform cells because they're pear-shaped neurons. Piriform cell layer, and you can see that layer also extends all the way under that molecular layer, all the way around that bend in the folium, and then here they are continuing on this other side. So these are really way cool neurons that you're going to learn about in neuroanatomy. They're very inhibitory neurons, and you'll learn that before you know it next semester. Underneath are a bunch of excitatory neurons. These are granule cells. They don't look like neurons, do they? Look like a bunch of oligodendrocytes, don't they? Well, they're not. They're granule cells. These are specialized neurons, little excitatory neurons. Interestingly, the most excited or hot rod neurons tend to be small neurons. The big ones tend to be kind of lazy. So these are granule cells and piriform cells. Okay, granule cell layer, and just like all the other layers, here it is, coming up, bending at the apex of that folium and coming back down. Those granule cells bend around a central core of axons. Here's our axons right here. Because white matter is deep, gray matter is superficial in the central nervous system. So here's our underlying deep white matter. And yes, there will be oligonuclei in the white matter because they are myelinating these axons in the white matter. In the gray matter, they are performing a supportive cell function to the neurons. So here is an oligo in the white matter right here and here, and it's supporting this nucleus of a neuron right here. Probably a basket cell, but you can't see him without special stains. All you can see is that neuron's nucleus. And let's see, one more thing and I'll cut you loose. Choroid plexus. Let's see, is there a choroid plexus in this section? No, I think I have to go back to this section. There we go. Another type of ependymal cell is the, another type of glial cell is the ependymal cell. They lined fluid-filled spaces. So this is the third ventricle. And here we have these ependymal cells lining that third ventricle. So they help form a barrier between the underlying nervous tissue and this fluid-filled space where we have cerebral spinal fluid flowing. These ependymal cells are ciliated. The cilia often become fairly damaged during processing. So here's some cilia here and here. They aren't the most beautiful cilia you've ever seen because again, they don't survive processing very well. These ependymal cells are very delicate. Now, these ependymal cells they line the ventricles, and in certain places along these ventricles, they'll fuse with the pia mater. Remember, I said the pia mater is highly vascular. So here we are. This is the pia mater. You can see that connective tissue right against the nervous tissue here. And here are the big distended blood vessels associated with that pia mater. These ependymal cells will fuse to some of these blood vessels right here, and these blood vessels will become modified. The walls will become fenestrated and sealed off by these ependymal cells, and these ependymal cells will pull plasma from the blood vessel, modify it, and secrete it into the ventricle. So where these ependymal cells fuse to the pia mater and form this special what do I want to say? It's like a tuft. Yeah, this special tuft of cells that protrudes into the ventricle. We call this a choroid plexus. Choroid plexus, and you'll see them grossly in neuroanatomy, but microscopically in histology, you'll also see them. So you'll want to recognize this. ID this as a choroid plexus. ID this modified ependymal cell layer here on the surface, ID the underlying connective tissue and blood vessels, which together is called the pia mater, and know what it does. Know that it secretes cerebral spinal fluid. These modified ependymal cells are not ciliated, but the ependymal cells lining the ventricles are ciliated, and they help form part of a barrier, a protective barrier between the nervous tissue and the ventricles where the cerebral spinal fluid flows. All right, that's it. Really, I've shown you everything you need to see today. So if you want to spend the rest of the time preparing for the quiz, we're okay with that. We will answer any question on anything. If you want to look at this material further this morning, we'll answer questions about this. If you want to ask questions about anything else, we're good with that too. And I think, Dr. K, you want me to hand it back over to you and you talk to them about
Sure.
what
Can you hear me?
Yeah, let me stop sharing here. Okay.
Okay, sure. And I guess what I'd like to say is just that we can we'll go into breakout rooms till about, what we'd like to do is go in breakout rooms till we'll go into one breakout room. We won't jump around today because it's a shorter period of time. And what we'll do is we will go ahead and at 9:10, I would like to have, I see that we have 121 students here. And what we're going to do is just come back to the main room leave your breakout room and go to the main room at 9:10. And the only reason why we want to do that is just to make it comfortable for if anybody has any questions, and we'll have a good 5 minutes or so to make sure that everybody can go ahead and get on to their quiz, whether they're in the classroom or whether they're in the actual remote site. And what I will do is I will go over at that point in time to the classroom so that the individuals that have class that are in the classroom can actually take that portion. I believe we have 35 of them currently downloaded that are going to take it in the classroom. We do have everybody downloaded, so I'm very, very happy that everybody was able to do that. So we have 125 downloads, which is wonderful. And so we'll come back at that point in time. So are there any questions from anybody who's listening? Okay, well then, if everybody is ready to either review or learn the nervous system and MALT and BALT, we will send the instructors to the breakout rooms and see you back at 9:10. Thanks, everybody.
I have a quick question.
Yes.
Oh,